Hello and happy Thursday. The plan today is to kind of go over some of the video stuff we did yesterday. I didn't get any questions, but I'm going to go through some stuff anyhow, and then talk in depth about something called transpiration. And there's going to be two things due today. It's a worksheet on transpiration rates, and then there's a virtual lab. This will be our first summative lab. That's a required IB lab. On your radar is a quiz tomorrow on flower structure and seed structure. And then coming up on Monday is that 6.6 test that we talked about. So first thing, if you haven't turned in your video questions, do so. And because we're doing twice as fast, we're doing two pre-classes today. So do pre-class day 3 and day 4. So pause, turn in your homework, and do your two pre-classes. Okay, I wanted to talk through the book. If you haven't looked at the book yet, if you don't have a book at home, there are some in the library that you can check out and bring home. Everything you need is in there, so as you have questions, we're in topic 9, and we're going to be covering transpiration first. Now, if you read through this, a lot of this stuff was in the videos yesterday, and so if the videos felt a little overwhelming, that's okay. We're going to go through them all again. So transpiration is today's topic. So my main video takeaways as I was jotting down notes here. So first, it was monocot versus dicot, and it just gave you really no background about it, but the fact that there are both types, and it just means one baby cot. The cot is a cotyledon, which is a baby, or two little babies in there, and then the plant features are different, and that was all that they kind of introduced. Not a huge thing to worry about yet. Next was the 3 types of tissues, and he did a good job of talking about how humans, we have 4 types, plants have 3 types, and they're broken down into dermal, which is like skin, and then there's ground tissue, which is a lot of the middle, the meat and potatoes of the plant, and then there's the vascular, which is the tubes, the transport, like our blood vessels. And so in this picture, the purple is the vascular tissue, and it looks different. The roots, it's in the middle. Stem, it's in like a circle, and then up in the leaves, it's spread out. So, and if you think about it, a leaf has the veins. That's its vascular, its tubes. The ground tissue is this green stuff. It's the meaty part of the plant, and then outside is the dermis, the skin, and it could include bark. If the plant has bark. He explained that the 3 enkymas, as they say, parenchyma, collenchyma, and sclerenchyma, and some good analogies to remember the difference. Potato is parenchyma, and this is the juicy, spongy part of the potato. Most of it's parenchyma tissue. Most of celery is collenchyma, which is sturdier. All right, but not as sturdy as sclerenchyma, which is like the gritty part of a pear. Not all of the pear, but you know how you get that grit, and some people hate it. So but that's the super tough tissue that is in the pear that gives it that vibe. So sclerenchyma is the toughest, the strongest. Then collenchyma is sturdy, and then parenchyma is the softest. Okay. Next, he talked about growth, and I liked it. That idea that if you put a nail into a tree and you wait, it'll be the same height because it grows at the tips, right? They're called apical meristem. A fancy way of saying like the tip growing. And in the shoot, it looks different. This is the meristem right here. It doesn't have anything to fight against, so it can just grow. And then the root has that cap. it's like a helmet, so it can burrow into the ground. But these are all little boxes of cells. This is each little box is a cell, and they're dividing and growing. And so that's the two ways that plants can grow up, and that was the primary. And then it can do the secondary, which he talked about tree rings, where it gets wider. So he talked about growth. Then this is what you probably want to focus more on, and because we're going to do a little quiz tomorrow, is just some common flower parts. So it went through that there's the male part, the men. That's the male, men. That's the has the anther on it. That has pollen, which is plant sperm. So yeah, if you're allergic to pollen, you're allergic to plant sperm. You can write that on forms if you want. And then the pistil is the term for the female part. And the stigma is the sticky part where the pollen lands. And then the pollen will go down to the ovary. It has petals, and then the green things that cover the flower before it blooms are called sepals. So that's some flower parts. Then I'm fascinated with this idea of how plants figured this out to be able to kind of have an embryo, but then feed it with this other one. So each pollen, each one has actually 2 sperm in it. And so it sends the tube down when it lands on there, and it sends both sperm. So one actually makes the baby plant, the other one fertilizes this other cell, this 2 cells, and they make the endosperm, which is the food around it. And so if you think about, if you're eating any sort of nut, most of it is going to be, I'm thinking like a peanut, right? You crack the shell and it has 2 parts because it's a dicot, and then that's all endosperm that you're eating. Delicious, right? And then the actual embryo is at one point of that. So as you go through that, you can think about all the different seeds that we eat, and you can start to be like, oh, okay, you're eating the endosperm. He did a good job of doing fruit or not a fruit. I thought that was funny too. Okay. Lastly, talked about evolution of plants and how we started with our non-vascular, non-tubed. So these are mosses; they're low to the ground, and it was really difficult. They had to have water to reproduce still, because again, they all came from algae, from the water. So, but then the first ones to not really need water were these non-seed vascular plants like ferns. And he talked about how they did make the spore and they drop it. He used fancy words, gametophyte and sporophyte, and we'll get there. But ferns came next, and then pine trees showed up, and they're called gymnosperms. But they use cones, and actually that's their reproductive part. So the actual hard pine cones, that was the female cones, and then there's now you can look at a pine tree and you can see that the squishier pine cones are the male ones. So that's kind of cool. And some of the adaptations that he explained for the fire. And then for the last 140 million years, flowering plants have been the best plant because they're the ability to have seeds, and then they use insects to co-evolve. Pretty impressive adaptation and those are called angiosperms. So they all have fancy names, but hopefully you can see the progression, and hopefully you got some cool things to kind of start us off on this unit from those videos. But yeah, don't worry, we're going to go through notes on each section and talk more about all of these things. All right, so today, if you're looking at the plan, we are going to be doing the factors affecting transpiration rates and the transpiration virtual lab. I might make the transpiration virtual lab due on Monday because we're having some flash issues. But let's hop over to the factors affecting transpiration. This is what it looks like. And if you have flash, you could go through it yourself. I'm assuming you don't. And so what Ms. Martineau did is she actually went through it on her computer and recorded herself doing the lab. And so what you can do is just click on this video link and then follow along with her. and pause it and fill out your form as you go. So go ahead and stop right now, do this factors affecting transpiration lab, and then come on back. Okay, so the second thing, this virtual lab, we'll at least get started on it, and then we'll see if we can get a video posted for how to finish the lab. So, all right, you can try clicking on this one and follow along. I'm gonna go through it here. I'm gonna restart. All right, so transpiration is plant sweat. We're gonna plants, the leaves, water leaves the leaves, and then sucks up from the roots. So when water leaves the leaves, is called transpiration. We're going to be looking at how to measure that and some of the features of what happens there. So water comes in the roots, it goes up the trunk all the way to the leaves and out. Xylem is the type of tissue that was in the video a little bit, and that's the tubes that water travels up to get there. For looking closer, we have our little plant cells, so each box is a cell, and we have our water molecules. And remember, they add adhesion to the walls of the plant, and they cohesion to each other. So it kind of like pulled in a line all the way up. Hydrogen bonding is what causes this feature, and that was from last year. We talked about it. So water molecules have a negative pole and a positive pole, so they're polar, and the negative and positive, the opposites, attract with each molecule. So that gives you these hydrogen bonds forming, and that's what causes the cohesion. So this is the leaf now at our water. Have gone all the way up to this point, and in the leaf there's actually a hole in the epidermis called a stoma, and that is how it gets breathing. So leaves have mouths pretty much, and it is able to breathe in carbon dioxide, which it needs, and breathe out oxygen as it does photosynthesis. The bad thing is that water leaves as well, which it wants water. Hold on, my dog. Okay, sorry about that. My dog was dreaming. Okay, it loses the water out that stoma as well as it does photosynthesis. All right, so let's look. So this first one, this is now the worksheet. You can be filling out. So what property of water is the fact that it grabs the walls of the xylem. So it's one of the adhesions, but in this one is adhesion, because cohesion sticks to itself. Adhesion sticks to other things. Water molecules pull each other. What property is that? That's the cohesion one. Co means together. And then 3. Place the following in order from highest water potential to lowest water potential. Water is flowing from a high to low, so water starts in the roots, so C. Then it's going to go into the tubes in the stem, and that was the xylem. And then it gets up to the leaves, and then lastly to the air outside. So CDAB. Awesome. So let's talk about these guard cells, these mouths. The stoma is the opening, the mouth, and they have guard cells around them, which look kind of like a mouth, like lips. And so they can open and close, kind of weird. But these are microscopic, so you can't like go up to a leaf and look at it and see these. And it does that using potassium ions. This is kind of the stuff in bananas. So when it wants it open, when it needs to breathe, it opens them, and then when it's done, it can close them. When it's open, it actually uses water to make them like fill up, and they go whoop, almost like thinking those balloons that have the curved balloons, right? And then if it wants to close them, it just deflates them with water, and they close up. Kind of cool. So potassium goes in, and that causes the water to leave. Potassium out, and the water follows it. So photosynthesis, I don't think this is one of your questions. I think we do have to label this. I didn't see that. I was down here. We did that one. We don't have to do the photosynthesis one, but we can talk about it. So plants take in water, they take in carbon dioxide, and they take in sun, and they make oxygen and sugar for us. So this is the whole goal here of photosynthesis, and the transpiration is when water leaks out through the stomata when they're exchanging gases. Okay. So this one, so transpiration is going to happen when its stomata are open. The stomata are open when it wants to do photosynthesis. So would there be more transpiration, more water loss through open stomata in light or dark? Well, if it's dark, it's not going to be doing photosynthesis, so it'll have its stomata closed. So the answer is light. 2. Which would result in a higher rate of transpiration? A humid environment or a dry? Well, now if it's dry, there's still light, so it's going to be open to do photosynthesis. And humid is still light, so it'll be open to do photosynthesis. So now which one would lose more water? Well, dry is going to lose more water because humid is water in the air. So, all right, 3. Which conditions would have higher rate? Breezy or still air? Well, if you've ever experienced breezy days, it dries things quickly, right? So if you need to dry something, you can put it by a fan. Or I remember sticking my soccer socks out the window and rolling them up in the window so they would dry. So windy is going to breezy is what they wrote. Breezy is going to actually dry it more and do more transpiration. And then last one, I already answered it here, warm. And this is weird, right? And it's interesting that, because what plants will do if it's too hot, they actually have a feature where they're like, it's too hot, although it's light out, I want to do photosynthesis, it's just not worth it. So they'll actually close if it's like desert hot. So that's kind of interesting for that. So at that point, I think we're close to done. We'll talk about potometers. We'll do 5 and 6, and then we'll pause. Okay. So a potometer, this little fancy gadget, is designed to measure the transpiration rate. And so what you have is a U-shaped pipette with water in it. You stick a plant cutting in there, and you seal that so no air can get out. And so now, as the plant sucks up water, and water will leave the leaves, you'll be able to measure on this side how much water has left. So potometers, they measure transpiration, and you measure it by seeing how much water it leaves. So some cool experiments that I'd love to be able to do maybe next year is you set these potometers up, and then you do a control, and you do a humid, you do a windy, and you do bright lights, and you can change temperature and just see what happens. And so what this virtual lab is going to do is going to go through those for you and give you some data on that and see what happens. So at this point, you should be done with this part. If not, go back up and look at these things and fill that all in. And I will hopefully be posting a lab link to Schoology, and you can watch the virtual lab. If not, then this will be due on Monday instead of tomorrow, and we'll figure it out on Friday then. Okay, have a good one. Thanks. Miss you guys. See ya.